For most of Earth's story, there were no animals, no eyes gazed across the horizon, no jaws opened, no skeleton held a body upright. The continents were largely bare stone, shaped by impacts and fire in a sky thick with carbon dioxide, nitrogen, and methane. The sun itself shone fainter than it does now, and yet liquid water gathered as oceans, while chemistry slowly crossed the threshold into biology. The earliest life appeared at least 3.5 billion years ago, leaving traces in ancient stromatolites. These beings were single cells, simple and without nuclei at first, surviving heat, radiation, and unstable seas. Entire ecosystems existed without a single creature that could crawl or swim in the way we understand. The oxygen we breathe today was not part of their world. When cyanobacteria learned to split sunlight, oxygen emerged as a waste product. It reacted with iron, staining the oceans and forming vast bands of rust within stone. Around 2.5 billion years ago, this gas accumulated in the air during what scientists call the Great Oxidation Event, altering the planet's chemistry and causing widespread die-offs among organisms unprepared for it. Later, between 700 and 635 million years ago, ice spread so extensively that glaciers may have touched the equator, while life endured beneath frozen surfaces and near hydrothermal vents. But how could a planet filled only with microscopic cells ever give rise to animals? What was it like to stand on continents without forests, without insects, without a single footprint? How did oxygen shift from poison to possibility? And why did life survive global ice and atmospheric upheaval when it could have vanished entirely? Tonight, we will drift gently across this vast stretch of deep time, guided by stone, chemistry, and patient evidence. But before we begin, I'd love to know where in the world you're listening from and what time it is there. It is always fascinating to think of these stories being heard across so many different skies. If you enjoy these gentle journeys into Earth's distant past, please do consider leaving a like and subscribing to the channel. Now, take a deep breath, let your shoulders soften, and together let us step into the longest silence our planet has ever known. Before there was stillness, there was fire. The young Earth gathered itself from drifting debris about 4.5 billion years ago, its mass drawn together by gravity until pressure and heat transformed it into a sphere of molten stone. The surface did not resemble land or sea, but a shifting ocean of magma, glowing beneath a sky thick with vaporized rock. Impacts were frequent and immense, remnants of planetary formation colliding with enough force to liquefy crust as soon as it tried to harden. Each collision reshaped the surface, carving basins, raising ridges, and delivering both destruction and new material from beyond. As the bombardment gradually eased, the outermost layers began to cool. A fragile crust formed, dark and unstable, floating atop deeper currents of heat. Volcanoes opened across the surface, releasing vast plumes of gas that gathered into an atmosphere unlike the one known today. Carbon dioxide dominated, mixed with nitrogen and traces of methane and water vapor. There was no breathable oxygen, no protective shield of ozone. The air would have felt dense and acrid, filtering a sun that shone fainter than it does in the present era. Even so, light reached the surface, touching rock that had only recently ceased to glow. Cooling continued over immense spans of time. Water vapor condensed as temperatures declined, falling first as steam, then as relentless rain. It poured onto hot stone, hissed back into vapor, and fell again. Gradually, as the surface stabilized further, liquid water began to remain. Basins carved by impacts and tectonic shifts filled, and the first oceans spread outward, dark and mineral rich. Though the sun emitted less energy in these early ages, greenhouse gases trapped enough warmth to prevent the planet from freezing entirely. Heat from within the Earth also rose through fissures and vents, sustaining warmth beneath the surface. The interaction between water and rock altered both. Minerals dissolved into the oceans, creating a complex chemical broth. Lightning crackled through thick clouds. Ultraviolet radiation poured down without resistance from ozone. Along volcanic coastlines and in the deep sea near hydrothermal vents, gradients of heat and chemistry formed natural laboratories. Iron, sulfur, carbon, and other elements cycled through reactions driven by energy from sunlight and from the planet's interior. The crust shifted slowly, plates beginning their long motion, recycling material from surface to mantle and back again. Within these restless environments, chemistry edged towards something more intricate. Molecules assembled, separated, and reassembled in patterns that gradually increased in complexity. Some structures proved more stable than others, persisting long enough to interact further with their surroundings. The oceans were not calm in a familiar sense, yet within their turbulence lay pockets where reactions could proceed undisturbed for critical intervals. Over spans of time difficult to fully grasp, networks of molecules capable of copying aspects of themselves emerged. The boundary between non-living chemistry and the first biology did not appear as a sudden event, but as a transition woven through countless cycles of trial and persistence. By degrees, the planet became less violent, impacts grew rarer, crust thickened, oceans deepened, and atmospheric conditions stabilized within limits that allowed these fragile chemical systems to endure. Earth had no footsteps, no movement across solid ground, yet beneath its waves and within its rocks, the foundations of life were taking shape, quietly preparing a transformation that would unfold over billions of years. In the quiet that followed fire and falling stone, the oceans became a cradle of minute persistence. By at least 3.5 billion years ago, something within those waters was no longer only chemistry drifting and dispersing, but life maintaining its delicate hold against a restless world. The evidence rests in ancient rock formations, known as stromatolites, layered structures built grain by grain as microscopic communities trapped sediment and bound it together. These stone records endure in places like Western Australia and South Africa, rising in low domes that carry within them the imprint of organisms too small to see without magnification. Their shapes speak softly of repetition, of growth guided by cycles of light and mineral flow. The planet at that time held no forests or reefs, no fins cutting through waves, no shapes casting shadows beneath the surface. Life existed only in the oceans, spread thinly through shallows and along submerged margins where sunlight could still reach. Every living thing was single-celled, each unit enclosed by a membrane, sheltering fragile molecules from the vastness outside. These earliest organisms lacked a nucleus, their genetic material floating freely within the cell, a simple architecture now described as prokaryotic. Yet simplicity did not mean weakness. Within those tiny boundaries, reactions unfolded with precision, drawing energy from available sources and channeling it into replication. Microbes came to dominate the planet because nothing larger had yet taken form. They coated sediments in thin films, drifted in open water, clung to mineral surfaces near hydrothermal systems. Some metabolized chemical gradients rising from the crust, thriving in darkness where sunlight never reached. Others depended on the faint illumination filtering through the upper layers of the sea. In environments where temperatures surged and fell, where ultraviolet radiation penetrated without the shield of ozone, these cells endured. Their enzymes functioned within wide ranges of heat. Their membranes adapted to fluctuating salinity and acidity. Survival did not arrive as a single innovation, but as countless small adjustments preserved across generations. The world they inhabited was still unsettled. Volcanic outgassing continued, altering the chemistry of air and water. Impacts, though less frequent than before, still disturb the surface. Oceans circulated minerals leached from rock, creating gradients that microbes exploited. Iron was abundant, dissolved in seawater and ready to react. Sulfur cycled through microbial metabolisms, linking the crust to the ocean in invisible exchanges. Each cell, microscopic and solitary, participated in transformations that slowly altered planetary systems through photosynthetic and chemosynthetic pathways that would diversify over time. Microbes reshaped their environment even as they depended upon it. Across immense spans of time, these single celled beings reproduced, divided, and spread. Mutation introduced variation. Selection favored configurations able to persist under prevailing conditions. There were no bodies composed of tissues, no organs coordinating function, only individual cells interacting in loose communities. Yet in their aggregate, they formed ecosystems, stabilizing sediments, influencing gas concentrations, and establishing feedback loops between biology and geology. The surface of the Earth, though silent in any familiar sense, was increasingly animated at the microscopic scale. Stromatolitic mounds continued to rise layer upon layer, recording seasonal rhythms and tidal pulses. In their lamination lies testimony to continuity, to life sustaining itself long before eyes evolved to witness it. For billions of years, microbes remained the primary actors upon the planetary stage, subtle yet transformative, weaving the earliest biological fabric within oceans that held no other forms. Long before air became something that could be breathed, it was something that transformed. Within shallow marine shelves where microbial communities gathered in dense films, a new metabolism began to unfold. Certain cyanobacteria developed the ability to use sunlight to split water molecules, drawing electrons from hydrogen and releasing oxygen as a byproduct. This innovation did not arise with ceremony. It emerged quietly among countless microscopic cells, each one pursuing energy in a world still ruled by chemistry and stone. Yet the consequences would extend far beyond the thin mats where it began. At first, the liberated oxygen did not linger in the atmosphere. It entered seawater already rich in dissolved iron, a metal abundant in its reduced form under earlier conditions. The oxygen bound readily with this iron, forming insoluble oxides that drifted downward and settled on the ocean floor. Layer upon layer accumulated over immense spans of time, alternating between darker bands, deposited under anoxic conditions, and red hues, marking pulses of oxidation. These sedimentary patterns hardened into what are now known as banded iron formations, vast geological archives of a world in chemical transition. Their stripes remain preserved in ancient cratons, testimony to reactions that unfolded long before complex life. For millions of years, this interplay acted as a vast sink, absorbing the oxygen produced by cyanobacteria. The oceans served as a buffer, capturing much of the gas and preventing it from building freely in the air. Gradually, however, the balance shifted. Iron supplies in many regions became saturated, and additional oxygen began to persist in seawater before diffusing upward. The atmosphere, once dominated by volcanic gases and reducing compounds, started to change. Around 2.4 billion years ago, concentrations rose sufficiently to mark what geologists describe as the Great Oxidation Event, a threshold in planetary history when oxygen became a stable and influential component of the sky. For organisms adapted to an anoxic environment, this shift was not benign. Oxygen is highly reactive, capable of damaging cellular components through oxidative stress. Many early microbes lacked protective enzymes, such as catalase and superoxide dismutase, that later evolved to manage reactive oxygen species. As oxygen levels increased, habitats that had long supported anaerobic life grew hostile. Widespread die-offs likely unfolded across oceans and sediments, though the fossil record preserves them only indirectly through chemical signatures. Entire microbial lineages may have retreated to isolated niches where oxygen remained scarce, such as deep subsurface environments and stratified waters. At the same time, new possibilities emerged. Oxygen enabled more efficient energy extraction through aerobic respiration, yielding far greater quantities of adenosine triphosphate from organic molecules than anaerobic pathways allowed. Though this advantage would take immense spans of time to reshape life's architecture, the biochemical groundwork had been established. The atmosphere's gradual oxygenation also contributed to the formation of an ozone layer high above the surface, reducing the intensity of ultraviolet radiation that reached shallow waters and coastal margins. The transformation was not abrupt in human terms, but unfolded across hundreds of millions of years, punctuated by fluctuations and feedback loops between biology and geology. Volcanic outgassing, burial of organic carbon, and weathering of continental rocks all influenced the pace and extent of oxygen accumulation. Yet, once initiated, the process altered the planet irreversibly. Iron chemistry shifted, methane concentrations declined, and global climate responded to the evolving balance of greenhouse gases. In this quiet revolution, a byproduct of microbial metabolism reshaped oceans, air, and the trajectory of life itself. Oxygen, once an accidental release from sunlit cells, became an enduring feature of Earth's surface environment, setting conditions that would govern every biological chapter that followed. There came an interval when sunlight touched the surface yet offered little warmth. Between 720 and 635 million years ago, the planet entered a succession of profound glaciations that altered its appearance and internal balance. Ice expanded from polar regions toward lower latitudes, brightening the globe as reflective surfaces replaced darker seas. With more incoming light returned to space, cooling intensified, reinforcing the advance. Over immense spans of time, frozen expanses grew until much of the ocean lay sealed beneath a rigid lid. Clues to this transformation remain embedded in ancient stone. Sedimentary layers, once deposited in tropical waters, contain fragments of rock dropped from melting ice, preserved far from the poles. Scratches etched into bedrock reveal the slow passage of glaciers across terrain that had rested closer to the equator in those distant ages. Carbonate deposits overlying glacial sediments display abrupt chemical shifts, suggesting dramatic swings in temperature and atmospheric composition. Together, these records indicate that ice may have extended across nearly the entire ocean surface, encasing waters that had long served as the principal domain of life. Beneath this frozen veneer, conditions were harsh yet not entirely still. Sea ice reduced exchange between atmosphere and ocean, limiting circulation and altering the distribution of dissolved gases. Surface waters cooled to near freezing, while deeper layers retained residual heat from the planet's interior. Hydrothermal systems along mid-ocean ridges continued to release minerals and warmth, sustaining localized pockets where chemical gradients persisted. In such settings, microbial communities endured, relying on metabolic pathways independent of direct sunlight. Other organisms survived within thin films of liquid water trapped between ice and sea, sustained by faint light filtering through translucent layers. While the surface remained locked in ice, Volcanic activity continued largely unseen. Tectonic forces carried crustal plates across the mantle, and eruptions released carbon dioxide into the atmosphere. Under normal conditions, silicate weathering would draw down this gas, but with continents buried under ice, that regulatory process diminished. Gradually, carbon dioxide accumulated, thickening the atmosphere with greenhouse potential. Over millions of years, the balance shifted as warming from trapped heat began to counteract the planet's high reflectivity. Ice margins retreated incrementally, exposing darker waters that absorbed more sunlight and accelerated the thaw. The transition from frozen dominance to open ocean was not instantaneous, but unfolded through feedbacks between radiation, chemistry, and circulation. As glaciers melted, they delivered vast quantities of ground rock into the sea, increasing nutrient availability. Weathering resumed on newly exposed continents, altering the composition of rivers and coastal waters. Carbonate minerals precipitated in unusual abundance, forming distinct cap carbonates that today mark the close of these glacial intervals. Ocean chemistry responded to the influx of materials, adjusting redox conditions and nutrient cycles in ways that would influence subsequent biological pathways. These severe glaciations reshaped climate systems and marine environments without extinguishing life entirely. Instead, they imposed constraints that favored resilience, preserving lineages capable of withstanding prolonged darkness and cold. When open water returned and atmospheric conditions stabilized, ecosystems reorganized within a changed framework of chemistry and circulation. The memory of global ice remained written in rock, a quiet testament to a world that had endured near planetary freeze and emerged altered, yet still alive within its hidden depths. Stone lay exposed beneath an open sky, unsoftened by roots or moss, uninterrupted by leaves. The continents of this era were expanses of bare rock, their surfaces shaped by tectonic uplift and erosion yet untouched by any living cover. There were no forests anchoring slopes, no grasses binding sediment, no lichens spreading in pale patches across stone. The land held only mineral and fracture, light and shadow moving across unbroken ground. Without plants to anchor and fracture rock, weathering unfolded at a different pace. Mechanical forces such as temperature shifts and the expansion of water within cracks gradually split stone apart, but the intricate chemical assistance that later life would provide was largely absent. There were no root systems exuding organic acids, no dense microbial communities forming thick soils in sheltered depressions. The transformation from solid rock to fine sediment was slower, governed by wind, sparse precipitation, and the steady abrasion of exposed surfaces. Soil, in the sense familiar to a later world, had not yet formed. There were no dark layers rich with organic matter, no networks of burrowing organisms to turn and aerate the ground. Instead, regolith accumulated thinly in places where rock had fragmented, only to be swept away by runoff during storms. When rainfall did occur, water moved swiftly across impermeable stone, carving temporary channels and carrying with it dissolved minerals. Rivers cut through terrain that had never felt the tread of a living limb, transporting chemical signatures directly from land to sea without the buffering influence of vegetation. These flows delivered nutrients to the oceans in raw form. Phosphorus, iron, and other elements leached from continental surfaces and entered marine systems with minimal interception. Coastal margins received pulses of sediment that spread outward, settling into basins and altering local chemistry. In the absence of stabilizing plant cover, shorelines shifted with each climatic fluctuation, and deltas formed and eroded in long, quiet cycles. Above this austere terrain, the atmosphere lacked a substantial ozone layer for much of this time. Ultraviolet radiation from the sun reached the surface with greater intensity than it would in later eras. On land, there were no organisms exposed to it, yet its presence shaped the boundary between ocean and air. Shallow waters near coastlines received elevated radiation, influencing which microbial forms could persist near the surface. The interaction between solar energy and mineral surfaces also contributed to chemical reactions across exposed rock faces, slowly modifying their composition. Wind traveled across continents without interruption from trunks or canopies. It lifted dust from dry basins and carried it across vast distances, depositing fine particles far from their origin. Mountain ranges rose and eroded in cycles measured over millions of years, their outlines stark against empty horizons. The silence was not absolute, for air still moved and water still flowed, yet there were no calls, no rustling leaves, no subtle tremors of rooted life responding to light. In this world of bare continents, the boundary between land and sea was sharp and elemental. The surfaces that would one day host forests and fields remained mineral plains, awaiting the gradual interplay of atmosphere, hydrology, and biology that would eventually soften their contours. For now, the continents stood austere and uninhabited, their exposed faces recording wind, rain, and sunlight in layers of stone, across spans of time measured not in years, but in the slow turning of the mantle. The continents themselves gathered and parted in vast, deliberate motion. Long before forests or footprints, immense landmasses converged to form a supercontinent known as Rodinia. Its assembly brought cratons together along sutures forged by mountain-building events, raising high ranges that would later erode into memory. The joining altered ocean circulation, constricted marine gateways, and reorganized the distribution of heat across the globe. Land that had once drifted apart now stood contiguous, reshaping wind patterns and the routes by which currents carried dissolved minerals. Rodinia did not remain whole. Tectonic forces operating beneath its broad expanse initiated rifting that slowly fractured the supercontinent. Crust stretched and thinned, volcanic activity traced new boundaries, and basins opened to admit water. As fragments drifted away from one another, coastlines lengthened, and new margins interacted with the sea. The breakup modified albedo and weathering rates, influencing atmospheric composition in subtle yet persistent ways. Each rearrangement of land shifted the balance between equatorial and polar regions, redistributing solar energy and altering patterns of precipitation. In the wake of Rodinia's fragmentation, continental pieces continued their motion, at times approaching and at others receding. Around 600 million years ago, many of these fragments came together briefly in a configuration called Pannotia. Its existence was transient in geological terms, a gathering that soon yielded to further dispersal. Yet even this temporary assembly influenced the surrounding oceans. When continents cluster, interior regions can become arid, while margins experience intensified weathering. When they separate, shallow epicontinental seas often spread across low-lying crust, creating expansive platforms where marine chemistry interacts closely with sediment. Continental drift reshaped ocean basins repeatedly during these ages. As plates moved, mid-ocean ridges generated new crust, and subduction zones recycled older seafloor into the mantle. The geometry of basins determined pathways for currents, which in turn regulated temperature distribution and nutrient transport. With more extensive shallow seas, sunlight penetrated broader areas, supporting microbial communities that depended on illumination. The expansion of continental shelves increased the interface between land and ocean, enhancing the delivery of ions derived from weathering and modifying the chemical backdrop against which life persisted. Climate responded to these rearrangements. When landmasses straddled equatorial regions, weathering could intensify under warm, wet conditions, drawing down carbon dioxide and exerting cooling influence. When continents clustered at higher latitudes, ice could accumulate more readily, reflecting sunlight and reinforcing lower temperatures. The position of continents also shaped monsoonal systems and the strength of ocean gyres, introducing feedback loops between tectonics and atmosphere. Over millions of years, these subtle adjustments accumulated into pronounced climatic shifts, recorded in isotopic ratios locked within carbonate and shale. Through all these transformations, the surface of the Earth never remained static. Mountain belts rose and wore away. Rift valleys widened into nascent oceans. Passive margins subsided beneath quiet seas. Each episode altered the physical template upon which future biological innovations would unfold. The interplay between lithosphere and hydrosphere created a dynamic mosaic of habitats, even in an era without animals. The supercontinents, before animals, were not merely arrangements of rock, but active participants in planetary evolution. Their assembly and dispersal regulated circulation, climate, and the flow of nutrients. In their motion lay a slow choreography that shaped the conditions of ocean and air, preparing a world whose geography was never fixed, only patient in its change. There is a quiet kind of threshold that does not announce itself with spectacle, only with consequence. During the Neoproterozoic, long after the first oxygen had altered the chemistry of sea and sky, atmospheric concentrations began to rise again in a gradual, uneven ascent. The increase was not smooth, but marked by fluctuations recorded in isotopic signatures and sedimentary layers, subtle traces of a world adjusting its internal balance. Over millions of years, oxygen moved from being a reactive participant in surface chemistry to a more stable and abundant presence in the air and upper oceans. This accumulation altered the limits within which cells could operate. Oxygen carries the potential for far greater energy yield through aerobic respiration than earlier metabolic pathways allowed. Where once life had depended largely on anaerobic processes, constrained by lower energetic returns, the expanding availability of oxygen widened biological possibilities. Cells capable of tolerating and exploiting this gas could extract more adenosine triphosphate from the same quantity of organic molecules, supporting increased internal complexity without immediate collapse under energetic strain. Within this shifting chemical landscape, eukaryotic cells became increasingly widespread. Unlike the earlier prokaryotic forms that dominated for billions of years, eukaryotes housed their genetic material within a defined nucleus, separating transcription from translation and permitting more intricate regulation of gene expression. Their cytoplasm contained internal membranes, compartments that specialized in distinct functions, reducing interference between metabolic processes. This architectural refinement did not emerge in isolation, but built upon earlier symbiotic events, most notably the incorporation of mitochondria. Mitochondria are thought to have originated from once free-living bacteria that entered into a stable partnership within a host cell. Rather than being digested, these endosymbionts persisted, contributing their capacity for oxidative phosphorylation to the composite organism. Over time, they transferred portions of their genetic material to the host nucleus, becoming indispensable components of cellular respiration. With mitochondria, cells could generate substantially greater energy output, sustaining larger genomes and more elaborate regulatory networks. The efficiency of ATP production increased the feasible size of cells and the diversity of functions they could perform. As oxygen levels continued their slow climb, particularly in surface waters, the ecological canvas broadened. Larger cells encountered fewer constraints imposed by diffusion alone, as internal energy supplies supported active transport and structural reinforcement. Cytoskeletal elements evolved to maintain shape and facilitate intracellular movement, allowing cells to grow beyond the minimal dimensions dictated by purely passive exchange. Some lineages began to experiment with adhesion and communication between neighboring cells, tentative steps toward cooperative arrangements that would eventually define multicellular life. The rise in oxygen also influenced redox conditions in the oceans, altering the distribution of dissolved metals and nutrients. In some regions, deep waters remained relatively low in oxygen, creating stratified environments that supported diverse microbial communities. Yet near the surface and along continental margins, expanding oxygenated zones provided new ecological gradients. The interplay between tectonics, weathering, and atmospheric composition continued to modulate these patterns, linking geological processes with biological opportunity. Energy availability does not compel complexity, but it enables it. With greater metabolic power, cells could afford to maintain additional genes, produce structural proteins, and manage more elaborate developmental programs. Over long spans of time, this energetic surplus allowed experimentation with size and form within the constraints of soft bodies and aqueous environments. The threshold was neither abrupt nor uniform across the globe, yet once crossed, it established conditions under which larger and more intricate organisms could persist, quietly setting the stage for further innovation within the oceans that remained, for now, the sole domain of life. Not all innovation arrives as a sudden leap. Sometimes it begins as proximity. In the long afterglow of rising oxygen, certain cells did not separate completely after division. They remained loosely attached, suspended together in the surrounding water, forming clusters that drifted as single units rather than solitary bodies. At first, these gatherings were simple colonies, each cell nearly identical to its neighbor, sharing nutrients and exchanging chemical signals through membranes that touched and flexed with gentle currents. Within these small assemblies, subtle differences began to emerge. Variation in gene expression allowed some cells to adopt slightly altered roles, adjusting metabolic rates or producing extracellular matrices that bound the group more securely. What had once been a collection of independent units gradually acquired a degree of coordination. Adhesion proteins strengthened connections between cells, reducing the likelihood that currents would pull them apart. Chemical gradients within the colony influenced behavior, guiding growth patterns that were no longer entirely symmetrical. Early algae exemplified this quiet transition. Photosynthetic eukaryotes, themselves descendants of ancient symbiotic mergers, began to form filamentous chains and simple sheets. Their bodies remained soft and flexible, composed of repeating cellular units lacking specialized tissues. Yet even within these filaments, rudimentary differentiation appeared. Cells along the outer surface engaged more directly with surrounding light and nutrients, while interior cells benefited from shared resources. Communication occurred through molecular signals passed across junctions, coordinating division and maintaining overall form. These early multicellular organisms did not possess organs or nervous systems. There were no centralized control structures directing movement, no muscles contracting in unison, no sensory arrays sampling the environment in complex ways. Instead, coordination arose from collective behavior, each cell responding to local cues that, in aggregate, shaped the entire body. Growth remained slow and largely dictated by diffusion and the availability of nutrients. Bodies expanded to sizes still modest by later standards, yet larger than single cells could easily sustain alone. Multicellularity offered advantages under certain ecological conditions. Larger size reduced vulnerability to predation once such interactions began to appear and permitted more efficient exploitation of light in the upper ocean. Clusters could sink more slowly or remain suspended more effectively, depending on their density and shape. Some lineages experimented with simple life cycles that alternated between single-celled and multicellular stages, preserving flexibility while exploring new organizational frameworks. The genetic toolkit required for these experiments did not arise from nothing. Genes involved in adhesion, signaling, and regulation had earlier functions within single cells, but were repurposed and elaborated to manage interactions among neighbors. Regulatory networks expanded, allowing cells within a colony to activate distinct subsets of genes. This incremental layering of complexity did not transform the oceans overnight. Instead, over millions of years, soft-bodied forms diversified into filaments, spheres, and flattened mats that clung to substrates or drifted in gentle suspension. Even as these multicellular experiments proliferated, the architecture of life remained delicate. Without hard parts, these organisms left limited direct fossil evidence, their impressions preserved only under exceptional circumstances. Yet, molecular clocks and rare sedimentary imprints suggest that by this time the groundwork for more intricate bodies had been established. Cells had learned to remain together, to communicate, and to distribute labor in primitive ways. In quiet waters where sunlight filtered down and nutrients circulated through slow currents, the first sustained attempts at building bodies from many parts continued, tentative yet persistent. Impressions in stone begin to appear with a new kind of presence, around 575 million years ago. Shallow outlines pressed into ancient seafloors and preserved beneath layers of sediment. These markings are neither ripple nor fracture, but the flattened signatures of organisms large enough to be seen without magnification. After billions of years dominated by the microscopic, the Ediacaran world introduced bodies measured not in microns but in centimeters, sometimes stretching the length of a human forearm, resting quietly upon or within marine sediment. These organisms were soft-bodied and largely without mineralized support. Their forms were often thin and quilted, composed of repeated segments or fronds branching from a central axis. They lacked hard shells, bones, or protective coverings that would later dominate the fossil record. Instead, their preservation depended on exceptional circumstances in which rapid burial shielded delicate tissues from decay. Fine-grained sediments draped across their surfaces, capturing outlines and subtle textures before decomposition erased internal detail. Many of these early macroscopic organisms lived anchored to the seafloor, attached either directly to sediment or to microbial mats that still carpeted portions of the ocean bottom. Others lay partially embedded, their flattened bodies conforming to the contours beneath them. There is little evidence for active movement in most forms, and no clear anatomical features that resemble mouths, digestive tracts, or sensory organs as recognized in later animals. Some may have absorbed nutrients directly through their outer surfaces, drawing dissolved organic matter from surrounding water. Others could have relied on symbiotic relationships with microorganisms, harvesting energy indirectly through partnerships established at cellular boundaries. The structure of these bodies hints at developmental programs more coordinated than earlier colonial experiments. Patterns repeat with mathematical precision, suggesting underlying genetic regulation that orchestrated growth along defined axes. Certain frond like organisms display branching sequences that echo natural fractals, maximizing surface area while maintaining stability in gentle currents. Radial forms spread outward in circular symmetry, distributing their tissues evenly across the sediment surface. Though lacking complex organs, these organisms demonstrate an architecture beyond simple aggregation, indicating that multicellularity had reached a new threshold of organization. The seafloor environment itself shaped their existence. Oxygen levels near the surface were sufficient to support larger bodies, yet deeper waters may have remained relatively low in concentration. Sediments held chemical gradients influenced by microbial metabolism, and subtle shifts in redox conditions could determine where these organisms thrived. Stability characterized many Ediacaran communities. Growth appears gradual, and ecological turnover seems slower than in later periods. There is no widespread evidence of predatory damage or rapid disturbance in early assemblages, allowing these soft forms to persist across expansive marine platforms. Though their affinities remain debated, the Ediacaran organisms represent the first sustained radiation of large, visible life. Some lineages may connect distantly to later animal groups, while others likely belong to experiments that left no direct descendants. Their bodies record a moment when biological complexity expressed itself openly upon the seafloor, no longer confined to microscopic scales. In quiet marine basins, beneath waters stirred only by tides and slow currents, the first macroscopic ecosystems unfolded, leaving delicate imprints that would endure as stone long after the organisms themselves had dissolved. On certain slabs of ancient sandstone, preserved beneath layers that once lay at the bottom of a quiet sea, there are forms that resemble nothing living today. They appear as low-relief impressions, their outlines crisp against stone that has held their memory for hundreds of millions of years. Among them are broad oval bodies such as Dickinsonia, segmented into repeating units that radiate from a central axis, each segment slightly offset from the next. The pattern is orderly yet unfamiliar, lacking any visible head, limb, or tail. There is no indication of eyes, no opening that clearly served as a mouth, no trace of internal organs discernible in the rock. Elsewhere in these same sediments lie the delicate imprints of frond, like organisms such as Charnia, rising from a single holdfast into branching arrays that divide again and again with quiet symmetry. Each branch mirrors the next in a self-repeating structure that resembles a fern yet bears no true leaves. The entire organism seems arranged along a central stalk, its surface subdivided into finer units that echo the larger form. This fractal organization maximized surface area, perhaps enhancing nutrient exchange in waters where resources were dissolved rather than captured through active feeding. Radial symmetry appears in several Ediacaran forms, bodies arranged around a central point rather than along the bilateral axis familiar in most later animals. Some resemble discs etched into sediment, their circular outlines segmented into petal-like divisions. Others take on more complex geometries, their patterns suggesting developmental programs, unlike those that shape modern animal embryos. The repetition of quilt-like modules across their bodies gives these organisms a textured appearance, as though stitched from living fabric and pressed gently into mud. Interpretation remains challenging. Certain chemical signatures preserved alongside Dickinsonia fossils indicate that they produced cholesterol like molecules, a trait associated with animal metabolism. This suggests at least some of these organisms may lie close to the ancestry of later animal lineages, yet many others resist classification. Their body plans do not align neatly with existing phyla, lacking the defining features that mark sponges, cnidarians, or bilaterians. They may represent experiments in multicellularity that diverged from the line leading to modern animals flourishing for millions of years before vanishing without descendants. These strange shapes occupied seafloors covered in fine sediment and microbial films, resting either upon the surface or embedded shallowly within it. Some Dickinsonia specimens appear to have shifted position slightly, leaving faint traces of movement across the sediment, though not in ways that suggest purposeful locomotion. Growth seems to have occurred by adding modules at one end, enlarging the body gradually over time. There is little evidence of predation in early assemblages, allowing such soft bodies to persist without defensive structures. The seas in which they lived were silent of sound in any familiar sense, stirred only by gentle currents and the slow settling of particles through water. Within that stillness, these organisms embodied a new expression of complexity, their forms broad and visible against the ocean floor. They demonstrate that life, once confined to microscopic dimensions, had begun to explore architecture on a larger scale, testing arrangements of tissue that defy simple placement within the tree of life. In their quiet diversity, the Ediacaran seas held shapes that would leave only impressions, enigmatic and enduring, in stone. The seafloor communities of this time reveal an absence that is as telling as any presence. In slab after slab, preserving delicate impressions of soft bodies, there are no clear bite marks, no gouges that indicate tearing flesh, no skeletal fragments shattered by force. The fossils rest intact within fine sediment, their outlines undisturbed by the violence that would later become common in marine ecosystems. For millions of years, life appears to have unfolded without active hunting, without jaws closing upon prey. In such ecosystems, energy flowed along quieter pathways. Many organisms likely absorbed dissolved nutrients directly across their outer surfaces, relying on gradients of organic molecules in surrounding water. Their flattened or frond-like shapes may have maximized surface area, increasing contact with nutrient-rich currents that drifted slowly over microbial mats. Others probably filtered suspended particles, drawing water across tissues capable of trapping microscopic food. Movement, where it occurred, seems limited to subtle repositioning rather than pursuit or escape. Without predation driving rapid adaptation, ecological competition may have remained comparatively low. Space on the seafloor was occupied, yet not fiercely contested in the manner seen in later eras. Communities display remarkable stability across extended intervals in the fossil record, suggesting that dominant forms persisted with limited disruption. Growth was measured and gradual. Bodies enlarged by adding modules or extending branches, not by racing towards speed or defensive complexity. The environmental backdrop supported this stability. Oxygen levels in surface waters were sufficient to sustain larger, soft-bodied forms, but not necessarily high enough to fuel intense metabolic activity associated with rapid movement. Currents moved sediment gently across broad shelves, redistributing nutrients without scouring the substrate. The absence of burrowing animals meant that microbial mats could blanket sediments uninterrupted, creating firm yet flexible surfaces upon which Ediacaran organisms could anchor. These mats may have contributed to preservation as well, stabilizing fine sediments that later captured delicate impressions. In ecosystems without active hunting, selection pressures emphasized endurance and efficient resource use rather than speed or armor. Morphologies optimized exposure to water flow, maximizing nutrient absorption while maintaining attachment in shifting sediments. Reproduction likely occurred through simple release of propagules into surrounding waters, dispersing offspring without elaborate courtship or territorial behavior. Genetic variation accumulated slowly within populations that faced relatively constant conditions over long spans of time. This was not a world devoid of interaction. Organisms influenced one another through shading, competition for dissolved nutrients, and the subtle alteration of local chemistry. Yet these interactions lacked the sharp escalation characteristic of predator-prey dynamics. There were no teeth requiring harder tissues in response, no escalating arms races producing spikes, shells, or toxins. Ecological networks remained comparatively simple, structured more by chemistry and physical gradients than by aggression. The calm of these communities cannot be equated with stagnation. Evolution continued, refining developmental programs and expanding morphological possibilities within soft tissues. The groundwork for more complex interactions was being laid at the genetic level, even if not yet expressed through active predation. For now, the sea floor remained a place where organisms rested upon sediments bathed in filtered light, drawing sustenance from currents that carried energy without the urgency of pursuit. In that prolonged quiet, life persisted through cooperation with its environment rather than conquest of it, inhabiting a chapter defined by stability and slow transformation. Beneath the surface of those ancient seas, chemistry arranged itself in patterns unfamiliar to later oceans. The water column was not uniformly mixed, nor evenly oxygenated from top to bottom. In many regions, particularly away from shallow margins, deep waters likely held far less oxygen than the surface layers touched by air and light. Stratification created boundaries within the sea itself, gradients of redox conditions that shaped which metabolic pathways could persist at different depths. Iron moved through these waters in greater abundance than it would in most later eras. In its reduced form, soluble and mobile, iron accumulated within deeper layers, where oxygen was scarce. When occasional mixing events carried this iron upward into more oxygenated zones, it reacted and precipitated, settling once again toward the seafloor. These exchanges did not occur as singular catastrophes, but as recurring processes woven into ocean circulation. Over time, they left geochemical signatures preserved in sedimentary rock, hinting at a world where dissolved metals played a more visible role in daily biochemical exchanges. Sulfur followed a cycle distinct from that of the present day. In oxygen-limited environments, sulfate-reducing microbes thrived, converting sulfate ions into sulfide as part of their metabolism. This sulfide could accumulate in deeper waters, sometimes combining with iron to form insoluble minerals that rained quietly onto the sediment below. In other settings, sulfur compounds were reoxidized near the interface of oxygenated and anoxic layers, creating a dynamic boundary where chemical energy became available to specialized microbes. These sulfur transformations influenced nutrient availability and shaped the distribution of life across vertical gradients in the sea. Across vast stretches of the seafloor, microbial mats extended like living carpets. These dense communities of bacteria and archaea bound sediments together, secreting extracellular polymers that stabilized fine grains. Layered structures formed as different microbial groups occupied distinct zones within the mat, arranged according to their tolerance for light, oxygen, and chemical compounds. Cyanobacteria often dominated upper layers where illumination was sufficient, while beneath them other microbes engaged in fermentation, methanogenesis, or sulfate reduction. The mat functioned as a miniature ecosystem, cycling elements internally while also interacting with overlying water. Nutrient cycling remained primarily in the hands of these microbial consortia. Nitrogen fixation converted inert atmospheric nitrogen dissolved in seawater into biologically usable forms. Ammonia and nitrate moved through microbial pathways, transformed by nitrifiers and denitrifiers, depending on local conditions. Phosphorus released from weathered rock entered marine systems and was assimilated into cellular structures before returning to sediments through decay. Without burrowing animals to disturb the substrate, these cycles operated largely within thin surface layers, their gradients maintained by diffusion and subtle water movement rather than active mixing. Deep basins may at times have become eugenic, enriched in hydrogen sulfide, creating environments inhospitable to many forms of complex life that would later evolve. Yet within these chemical extremes, microbial diversity flourished. Adaptation to shifting iron, sulfur, and oxygen regimes allowed communities to occupy niches defined by chemistry rather than physical structure. The ocean before animals was thus not empty of interaction, but deeply structured by invisible exchanges among dissolved elements and microscopic metabolisms. In this chemically textured world, every gradient represented both constraint and opportunity. The interplay between stratified waters, reactive metals, and living mats established a foundation upon which later ecological transformations would unfold. For now, the rhythms of the sea were governed less by movement and more by redox balances, subtle currents, and the persistent labor of microbes weaving elemental cycles through quiet depths. Light crossed the surface of the ocean each day without being seen. It entered the water in long, shifting bands, scattering into blues and greens as it descended, touching microbial films and soft bodies that possessed no eyes to register its arrival. In this era, vision had not yet evolved. There were no lenses focusing images, no photoreceptor arrays translating brightness into pattern. Illumination existed as energy, not as sight. Yet the absence of vision did not render light irrelevant. Many microorganisms remained exquisitely sensitive to its presence. Photosynthetic cells responded to the angle and intensity of incoming sunlight, adjusting pigment concentrations and metabolic rates according to availability. Their internal chemistry followed cycles tied to the rising and setting of the sun, rhythms encoded within molecular feedback loops that oscillated across roughly 24 hours. Even without awareness, there was coordination between external light and cellular function, a quiet synchronization that linked metabolism to the planetary rotation. For multicellular forms resting upon the seafloor, light influenced distribution and growth without requiring perception in any conscious sense. Organisms occupying shallow marine platforms positioned themselves where illumination could support photosynthetic symbionts or enhance nutrient gradients shaped by temperature differences between surface and deeper waters. As depth increased and light diminished, communities shifted accordingly, each layer of the ocean defining a boundary for certain lineages. These transitions were guided not by sight, but by biochemical tolerance and energy acquisition strategies refined over generations. Movement in such a world remained gradual. There was no rapid darting in response to visual cues, no sudden pursuit triggered by the detection of prey. Without eyes to detect approaching threats or opportunities at a distance, Selective pressures did not favor high-speed locomotion. Bodies remained soft and often anchored, designed to maximize exchange with water rather than to chase or flee. Diffusion and gentle currents transported nutrients, and organisms responded primarily to immediate chemical conditions rather than distant visual signals. Day and night continued their alternation regardless of perception. With each rotation of the planet, light levels rose and fell, influencing temperature gradients in surface waters and subtly altering vertical mixing. Microbial communities adjusted enzyme activity and replication timing to these cycles, dividing or entering quiescent phases according to internal clocks entrained by illumination. Even cells living deeper in the water column could experience indirect effects as surface productivity waxed and waned, influencing the flux of organic material drifting downward. Ultraviolet radiation, moderated by the developing ozone layer, yet still significant, imposed further constraints. Certain pigments evolved to absorb and dissipate harmful wavelengths, protecting delicate molecular structures. In shallow environments, this filtering effect shaped which microorganisms could thrive closest to the surface. The spectrum of sunlight thus acted as both resource and hazard, guiding evolutionary paths without any organism needing to interpret an image of its surroundings. Life without eyes was not life without order. Patterns of growth, reproduction, and distribution responded to predictable cycles of light and darkness embedded within the turning planet. Cellular networks sensed intensity changes through photochemical reactions, initiating cascades that adjusted internal states. Over immense spans of time, these biochemical sensitivities would provide raw material for more complex sensory systems. For now, however, the oceans remained places where light governed processes invisibly, where energy flowed from star to cell without passing through perception, and where existence unfolded in gradients rather than in vistas. The planet was not silent, but nothing listened. Air moved across exposed continents, sweeping over stone and into shallow basins, its passage marked only by the shifting of fine grains and the low hum of motion against mineral faces. Winds gathered strength over open expanses where no vegetation interrupted their flow, circling mountain ranges and descending into valleys that held no creatures to stir in response. The atmosphere carried pressure and vibration, yet there were no ears to convert these oscillations into experience. Along the margins of the sea, waves folded and unfolded in steady rhythm. Tides rose and fell under the gravitational pull of the moon, advancing across microbial mats and retreating once more, leaving thin films of water that shimmered briefly before evaporating or draining back toward deeper channels. The impact of water against rock produced resonance, subtle percussion repeated through countless cycles. Storms churned surfaces into white turbulence and then subsided, restoring the slow undulation that characterized calmer intervals. These sounds existed as mechanical energy, traveling through air and water without interpretation. Beneath the surface, ocean currents traced invisible pathways shaped by temperature gradients and salinity differences. Dense waters descended while warmer layers drifted above, establishing circulation patterns that redistributed heat and dissolved nutrients. Turbulence generated low-frequency vibrations that propagated through the water column. Seafloors recorded the gentle settling of particles and the distant rumble of submarine landslides, yet no organism possessed specialized structures to detect these movements as discrete signals. The sensory world remained chemical and tactile rather than auditory. Volcanic activity added its own textures to the planetary soundscape. Eruptions fractured crust along tectonic boundaries, releasing gases and molten rock with force that shook surrounding terrain. Submarine volcanoes extruded basalt into dark water, creating plumes of heated fluid that expanded and cooled. Seismic waves traveled through solid earth, reflections of deep processes far below the seafloor. These vibrations passed through strata that would later cradle more responsive life, but at this time they encountered only stone and microbial communities insensitive to pressure fluctuations beyond immediate mechanical effects. On land, rainfall struck bare rock, producing sharp, transient echoes that faded quickly into open air. Rivers, unconfined by roots or soil, rushed across exposed surfaces during periods of precipitation, their flow carving channels and transporting sediment toward the sea. The absence of plants meant no rustling leaves, no trunks creaking in shifting winds. There were no footfalls, no calls rising at dusk, no wing beats interrupting stillness. Movement across the surface was driven by water and air rather than by muscle and intent. In shallow marine settings, currents brushed against soft-bodied organisms resting upon sediment. These interactions may have influenced orientation or growth patterns, yet without auditory organs, there was no perception of sound as a distinct dimension. Vibrations were part of the physical environment, integrated into fluid dynamics and substrate stability rather than translated into information. Communication among organisms occurred through chemical gradients and direct contact, not through pressure waves traveling at speed. The Earth of this era carried a constant murmur of geophysical activity, an orchestra without audience. Atmospheric circulation, oceanic convection, and tectonic adjustment produced a complex field of vibration extending from surface to mantle. Within that field, life persisted unaware of resonance in any conscious sense. The planet moved, water flowed, and stone fractured, while the absence of ears ensured that sound remained a property of matter alone, not yet a medium for behavior or exchange. As the great ice sheets retreated and brighter oceans reemerged, the planet did not settle immediately into balance. Instead, it entered a period of climatic instability shaped by the lingering effects of glaciation and the slow accumulation of greenhouse gases. Carbon dioxide, released continuously by volcanic outgassing while weathering processes had been suppressed beneath ice, now lingered in elevated concentrations within the atmosphere. With vast reflective surfaces diminished, solar energy was absorbed more readily by dark water and exposed rock, amplifying warming across continents and seas. Temperatures rose unevenly. Regions that had once been locked beneath thick ice experienced accelerated melting, feeding pulses of fresh water into the oceans. This influx altered salinity patterns, influencing density-driven circulation and reshaping currents that distributed heat around the globe. Coastlines migrated inland as sea levels climbed, drowning low-lying continental margins and expanding shallow marine environments. Newly flooded shelves became dynamic interfaces between land-derived nutrients and marine chemistry, areas where biological communities adjusted to shifting boundaries. High carbon dioxide levels sustained a greenhouse climate, trapping heat within the lower atmosphere and intensifying evaporation over open water. Precipitation patterns responded, sometimes increasing weathering rates on exposed rock and drawing down carbon through chemical reactions that sequestered it in carbonate minerals. Yet these feedbacks did not operate smoothly. Variations in tectonic configuration, ocean circulation, and biological activity produced oscillations in atmospheric composition and temperature that unfolded over millions of years. The system sought equilibrium but encountered continual perturbation along the way. Within the oceans, oxygen concentrations fluctuated in response to these changes. Warmer surface waters can hold less dissolved oxygen, and stratification may have intensified as temperature differences between layers grew more pronounced. In some basins, reduced mixing limited the transport of oxygen to deeper waters, fostering conditions where anoxia could develop intermittently. Geochemical records reveal shifts in redox-sensitive elements that point toward episodes of expanding low oxygen zones, followed by periods of partial recovery as circulation patterns adjusted. These fluctuations influenced marine ecosystems in subtle ways. Organisms accustomed to relatively stable conditions found themselves navigating gradients that shifted across time. Communities reassembled along continental shelves as sea levels rose and fell, tracking suitable oxygen levels and nutrient availability. Sediment deposition reflected alternating phases of productivity and restriction, preserving in layered rock the imprint of climatic pulses that reverberated through water and air. Meanwhile, chemical weathering intensified on continents newly exposed after glacial retreat. Silicate rocks reacted with atmospheric carbon dioxide in the presence of water, gradually drawing down greenhouse gases and tempering extreme warmth. Rivers transported increased loads of dissolved ions to the sea, altering alkalinity and promoting carbonate precipitation. These processes provided long-term stabilizing influences, yet they operated on scales measured in hundreds of thousands to millions of years, allowing interim periods of imbalance. The climate of this era can thus be understood as a series of adjustments rather than a fixed state. Post-glacial warming initiated cascading responses across atmospheric composition, sea level, and ocean chemistry. Carbon dioxide levels rose and declined in cycles linked to tectonics and weathering, while oxygen concentrations in marine basins expanded and contracted according to circulation and biological demand. In this shifting environment, life persisted within boundaries that were neither constant nor entirely predictable, adapting gradually to a planet whose temperature and chemistry were still finding new alignments after the long reign of ice. Across broad continental shelves where sunlight filtered through clear water, entire communities rested directly upon the sediment, fixed in place and arranged like living mosaics. The seafloor of this era was not churned or overturned by burrowing creatures. Its surface remained layered and largely undisturbed, a stable substrate upon which soft-bodied organisms could anchor themselves for long spans of time. Attachment occurred through holdfasts, discs, or simple basal surfaces that pressed gently into fine grains, securing bodies against the slow drift of currents. Beneath and between these larger forms, microbial mats continued to form cohesive films across the sediment. These mats functioned as both structural support and primary producers, converting light and dissolved nutrients into organic matter that entered the surrounding ecosystem. Their upper layers trapped particles carried in from nearby shores, while deeper layers facilitated anaerobic processes that recycled elements within tight biochemical loops. The mats stabilized the sediment surface, creating a firm yet flexible foundation that resisted erosion under moderate flow. Organisms dwelling upon this living carpet derived sustenance in ways suited to still environments. Some absorbed dissolved compounds released by microbial activity, relying on diffusion across thin tissues. Others may have hosted symbiotic microbes within or upon their bodies, integrating external productivity into their own metabolism. Filter feeding, if present, likely operated at low velocities, with water passing gently across surfaces designed to capture suspended particles without requiring swift movement. Energy flow through these systems was continuous but measured, lacking the bursts associated with active predation. Growth rates appear to have been slow when compared to later marine faunas. Fossil assemblages show individuals reaching substantial size relative to earlier forms, yet doing so through incremental addition of segments or expansion of branching structures. Without intense competition for space, or the threat of grazers consuming mat surfaces, communities could persist with little disturbance. Recruitment of new individuals may have occurred sporadically, filling available gaps gradually rather than in rapid succession. Minimal burrowing activity preserved clear stratification within sediments. Oxygen penetration likely remained confined to upper millimeters, with anoxic conditions prevailing just below the surface. This sharp chemical boundary supported distinct microbial zones and influenced the types of larger organisms that could inhabit particular niches. Sediment layers remained laminated, recording seasonal variations and subtle shifts in environmental conditions. The absence of deep mixing meant that nutrient exchange between sediment and overlying water was governed primarily by diffusion and minor bioturbation rather than active excavation. Spatial organization within these seafloor ecosystems suggests patterning influenced by light availability, water depth, and substrate stability. Frond-like forms often oriented themselves relative to prevailing currents, optimizing exposure to nutrient flux. Radial organisms occupied patches where sediment consistency allowed even distribution of weight. Microbial mats filled spaces between anchored bodies, ensuring that little bare substrate remained uncolonized. Over time, this arrangement created communities with a calm, internal rhythm, sustained by predictable exchanges between light, chemistry, and biological demand. The seafloor of this period functioned as a quietly integrated system. Attachment, rather than mobility, defined existence. Stability, rather than rapid turnover, characterized community structure. Layers of life rested upon layers of sediment, each influencing the other through chemical gradients and physical support. Within these expansive marine platforms, ecosystems operated without the turbulence of burrowing predators or the disruption of grazing herds. Instead, they persisted as long-lived assemblages anchored to a substrate that itself remained largely undisturbed, a foundation upon which later transformations would gradually build. In certain layers of late Precambrian sediment, the stillness begins to blur. Instead of only the pressed outlines of resting bodies, there are faint linear markings that cut across the surface, shallow grooves and winding tracks, preserved where soft sediment once held their shape. These trace fossils do not reveal the anatomy of the organisms that made them, yet they record behavior. For the first time, the seafloor bears evidence that something moved with intent, however simple that intent may have been. The trails are modest, often narrow and gently curving, sometimes looping back upon themselves. They lack the complexity of later burrow systems, yet they differ from marks produced by drifting debris or shifting currents. Their patterns suggest repeated contact between a body and the sediment surface, as though an elongated form pressed lightly against the substrate while advancing. In some cases, the track appears to undulate, hinting at muscular contraction along a soft body. The organisms responsible were likely small and lacking hard parts, perhaps worm-like in general form, though not necessarily related to modern annelids. Movement in this context did not mean rapid locomotion across vast distances. It may have involved slow gliding over microbial mats, or shallow probing into the uppermost sediment in search of organic matter. The substrate itself began to change as a result. Even slight disturbance disrupted the laminated structure maintained by microbial communities, allowing oxygen to penetrate marginally deeper and altering redox gradients just below the surface. These small modifications had outsized consequences for chemical exchange between sediment and water. The appearance of trace fossils indicates a shift in ecological strategy. Rather than remaining anchored and absorbing nutrients passively, some organisms began to explore their surroundings physically. Locomotion allowed access to new patches of organic material, increasing the efficiency of resource acquisition in environments where dissolved nutrients may have become more limited. With mobility came new selective pressures, including the need for coordination of muscular contractions and spatial orientation within a three-dimensional environment. Although the trails are simple, they mark the beginning of substrate disturbance, a process that would eventually transform marine ecosystems. Bioturbation, even at its earliest stages, broke the stability of the seafloor surface. Sediment layers that once accumulated in thin, undisturbed sheets began to mix slightly, blurring fine laminations that had persisted for millions of years. Microbial mats, long dominant in structuring the interface between water and sediment, faced incremental disruption as moving bodies crossed and grazed upon them. These first signs of movement did not erase the calm character of Ediacaran communities. Many organisms remained attached or sedentary, and predation was still minimal or absent. Yet within the quiet environment, a new dynamic had entered. Energy was now being invested in locomotion rather than solely in growth or surface exchange. Muscular tissues, however primitive, coordinated contractions that translated chemical energy into displacement across sediment. The record of these movements is delicate and incomplete, preserved only where rapid burial captured impressions before they faded. Even so, their presence signals a fundamental change. The seafloor was no longer only a place of rest and passive exchange. It had become a surface to traverse, a medium through which bodies could leave marks of passage. In the gentle arcs and subtle furrows etched into ancient mud, the first steps toward an animated world are written, understated yet irreversible in their implication. Size is not only a matter of shape, but of chemistry. As atmospheric and shallow ocean oxygen concentrations continued their gradual rise, the physical limits that had long constrained living cells began to ease. Oxygen serves as a final electron acceptor in aerobic respiration, enabling far more energy to be extracted from organic molecules than anaerobic pathways allow. With greater availability of this gas in surrounding water, organisms could sustain higher metabolic rates without exhausting their internal reserves. Energy budgets expanded, and with them, the potential to build and maintain larger bodies. For soft-bodied organisms lacking specialized circulatory systems, oxygen entered tissues primarily by diffusion. This process relies on gradients, with molecules moving from regions of higher concentration to lower concentration across membranes. Diffusion is effective over short distances but becomes inefficient as size increases, because internal cells lie farther from the external environment. In earlier periods, when oxygen concentrations were lower, these limits imposed strict constraints on how thick or voluminous a body could become without risking internal hypoxia. Cells deeper within a tissue might not receive sufficient oxygen to sustain metabolism. As oxygen levels rose, diffusion could support slightly greater distances before becoming limiting. Soft tissues could thicken modestly, and organisms could expand their surface area relative to volume through branching or flattening while maintaining internal function. The geometry of many Ediacaran bodies reflects this balance. Broad, quilted forms and frond-like structures maximized surface exposure, allowing oxygen to permeate tissues efficiently. In environments where shallow waters were well oxygenated, these strategies permitted growth beyond the microscopic scale that had characterized life for billions of years. Metabolic demands also shaped the trajectory of body size. Larger organisms require more energy, not only for maintenance, but also for growth and reproduction. Increased oxygen availability supported higher rates of ATP production within mitochondria, allowing cells to sustain more active biosynthesis and structural complexity. Proteins involved in cytoskeletal organization, intercellular adhesion, and extracellular matrix formation could be produced in greater quantities without overwhelming the organism's energetic capacity. This biochemical surplus provided room for developmental experimentation. The relationship between oxygen and size was not linear or uniform across all habitats. Deep waters may have remained comparatively low in oxygen, preserving constraints that limited growth in those regions. Stratification could create zones where only the upper layers supported larger bodies, while deeper communities retained predominantly smaller forms. Geochemical proxies suggest that oxygenation occurred unevenly across basins, meaning that ecological niches varied in their potential for size increase. Organisms occupying well-oxygenated shelves had access to different possibilities than those in more restricted settings. Even with enhanced oxygen, diffusion alone could not support unlimited expansion. Without internal transport systems such as blood vessels or specialized respiratory structures, there remained a threshold beyond which cells could not be adequately supplied. Thus, early increases in body size were modest relative to later animal forms equipped with circulatory networks. Nevertheless, the incremental enlargement seen during this interval signals a loosening of earlier energetic constraints. In quiet marine platforms where oxygen concentrations crossed critical thresholds, soft-bodied organisms grew broader and more architecturally complex. Their dimensions were a direct reflection of atmospheric and oceanic chemistry, an embodiment of the invisible gradients that sustain them. Through the interplay of diffusion limits and metabolic capacity, the physical scale of life expanded cautiously, guided by the availability of a gas that had once been rare and hostile, but now quietly underwrote new possibilities of form. The outline of the world was never fixed. Even as life explored new forms within the oceans, the continents themselves continued their patient migration across the mantle, driven by currents deep within the planet's interior. Plates shifted by increments too small to notice in a single lifetime yet immense when measured across millions of years. Land masses that had once converged began to fragment further, pulling apart along rift zones where crust thinned and basalt rose from below to create new seafloor. The geometry of the globe adjusted without haste, but never without consequence. As fragments separated, shallow seas expanded across broad continental margins. Newly formed ocean basins admitted water that flooded low-lying regions, creating extensive epicontinental seas that spread like luminous shelves beneath the sun. These environments were neither deep abyss nor exposed land, but transitional zones where light reached the seafloor and currents circulated nutrients with relative consistency. Their breadth increased the interface between rock and water, encouraging chemical weathering on emergent land and nutrient input along submerged edges. With the widening of shallow seas came diversification of habitat, variations in depth, substrate type, and water chemistry multiplied across continental margins. Some regions hosted fine sediments deposited in calm conditions, while others experienced stronger currents that sorted grains and sculpted ripple fields. Temperature gradients emerged between equatorial shelves and higher latitude basins, shaping biological distributions according to tolerance and energy supply. Oxygen concentrations could vary between enclosed inland seas and more open marine settings, producing distinct ecological niches even within broadly similar geographic zones. Fragmentation of larger continental assemblies altered atmospheric circulation as well. The distribution of land relative to ocean influences wind patterns, precipitation, and evaporation. When landmasses lie closer to equatorial regions, increased rainfall can intensify weathering, drawing carbon dioxide from the atmosphere and contributing to climatic regulation. When continents cluster at higher latitudes or fragment into dispersed islands, ocean currents may reorganize, modifying heat transport across hemispheres. These slow adjustments reverberated through climate systems already in flux from earlier glaciations and greenhouse intervals. Tectonic activity also generated new coastal topography, including uplifted margins and subsiding basins. In areas where crust stretched thin, rift valleys filled with seawater and evolved into narrow seaways, fostering localized circulation patterns distinct from open ocean gyres. Hydrothermal systems along spreading ridges continued to release minerals into surrounding waters, enriching certain habitats with chemical energy. The interplay between tectonics and ocean chemistry created a mosaic of environments within which life could experiment. For marine communities anchored to sediment or capable of limited movement, expanding shelves provided opportunity for dispersal. As habitats proliferated, lineages encountered new combinations of temperature, salinity, and nutrient availability. Some remained confined to stable niches, while others extended their range across adjacent platforms. The physical fragmentation of continents thus translated into ecological fragmentation and innovation, not through abrupt upheaval, but through gradual reshaping of available space. Continental drift did not act alone in shaping this transformation. It operated in stage upon which the next chapters of biological complexity would unfold, expanding shallow seas into vast arenas of quiet possibility. In the slow layering of sediment and the widening of shallow seas, something subtle began to tighten within marine communities. Organisms that had long coexisted in relative stability now encountered one another more frequently across expanding shelves. As habitats diversified and population densities increased in favorable zones, biological interactions grew more intricate. No sudden rupture marked this change. Instead, it unfolded as a gradual thickening of relationships among living forms. Where microbial mats once dominated open surfaces with minimal interference, larger soft-bodied organisms increasingly overlapped in space and influence. Fronds shaded adjacent tissues, altering light exposure at the scale of centimeters. Broad quilted bodies intercepted nutrient-laden currents, reducing the flux available to neighbors downstream. Even in the absence of active predation, such proximity fostered competition over dissolved resources and favorable attachment sites. Energy entering the system through photosynthesis and chemical pathways remained finite, and its distribution shaped success within local assemblages. Nutrient competition likely intensified as populations expanded in oxygenated shallow waters. Organisms reliant on diffusion for sustenance experienced gradients that varied subtly from one patch of sediment to another. Slight advantages in surface area or positioning relative to currents could determine growth rates over extended intervals. Genetic variation within lineages provided opportunities for differential success, favoring morphologies that maximized exposure to flow or optimized metabolic efficiency under fluctuating conditions. This quiet sorting did not generate dramatic turnover, yet it seeded ecological differentiation. Environmental variability added further complexity. Climate oscillations, though measured in geological time, influenced sea level and temperature patterns that reshaped coastal platforms. Shifts in salinity from freshwater input or evaporation altered chemical balances within semi enclosed basins. Oxygen concentrations in certain regions may have risen and fallen with circulation changes, creating pulses of constraint and release. Communities accustomed to stable conditions encountered new gradients, requiring flexibility and physiological tolerance. Interactions extended beyond direct competition. The presence of attached organisms modified local hydrodynamics, creating microhabitats in their lee where finer sediments accumulated or where nutrient concentrations differed slightly from surrounding waters. Such structural effects could facilitate colonization by additional forms, increasing complexity without overt aggression. Meanwhile, early mobile organisms that left faint trails across sediment began to influence mat integrity, redistributing organic matter and altering microbial communities beneath the surface. As these interactions accumulated, ecological networks became more interconnected. The outcome of growth for one lineage increasingly depended upon the configuration of neighbors and the state of surrounding chemistry. Energy flow through the system grew less uniform, channeled through multiple pathways shaped by position and morphology. In environments where shallow seas spread across fragmented continents, variability in substrate and water movement created a patchwork of selective pressures, encouraging local adaptation. This rising tension did not yet express itself as widespread predation or defensive escalation. Instead, it manifested as incremental shifts in dominance and distribution. Some morphologies flourished under new conditions, while others retreated to narrower niches. The overall pace of change remained measured, but beneath the surface, calm, evolutionary trajectories began to diverge more noticeably. The seafloor communities, once defined primarily by stability and gentle exchange, now bore the imprint of increasing interdependence and competition, subtle forces building within ecosystems that were gradually moving toward greater dynamism. The first burrows do not appear as dramatic tunnels carved deep into the earth, but as subtle distortions within once pristine layers of sediment. Where earlier seafloors preserved fine lamination with remarkable clarity, these newer deposits show disruption, small cylindrical passages, and gently curved shafts that penetrate downward a short distance before fading. The texture of the substrate begins to change, its surface no longer uniformly bound by cohesive microbial films. Something beneath the water has started to move within the sediment itself. These early burrowers were likely small, soft-bodied organisms capable of pushing through loose grains by muscular contraction and expansion. Their bodies would have lacked hard parts, yet they possessed sufficient strength and coordination to displace sediment as they advanced. The depth of their penetration remained modest at first, limited to upper layers where oxygen from overlying water could still diffuse. Even so, this behavior introduced a new form of ecological engineering. Sediment was no longer a static boundary between water and chemistry. It became a medium actively rearranged by living bodies. As burrows opened and closed, oxygen began to penetrate deeper into the substrate. Water carried dissolved gases into spaces created by moving organisms, altering redox conditions that had long persisted beneath undisturbed mats. Chemical gradients once maintained in sharp, narrow layers became blurred. sulfide-rich zones mixed with more oxygenated material, influencing microbial metabolism at the interface. Nutrients trapped within sediment were released into overlying water or redistributed laterally, changing the availability of resources for both microbes and larger organisms. The decline of extensive microbial mats followed gradually. These cohesive films depended on stable surfaces and minimal disturbance to maintain their layered structure. Repeated burrowing activity broke apart their continuity, fragmenting communities that had dominated seafloors for vast intervals. As mats thinned or retreated to niches less affected by excavation, sediment became more dynamic. Fine laminations grew less common, replaced by textures indicative of bioturbation. The physical appearance of the seafloor shifted from flat expanses to subtly pitted landscapes marked by openings and infill. This transformation carried implications beyond surface structure. By mixing sediment layers, burrowers accelerated nutrient cycling between deeper deposits and the water column. Organic matter buried within upper layers was exposed to oxygen, promoting decomposition pathways different from those favored under strictly anoxic conditions. Carbon and sulfur cycles responded accordingly, their fluxes influenced by the mechanical stirring of once stable strata. The ecological consequences extended outward from each small tunnel. For organisms inhabiting the seafloor surface, burrowing introduced both challenge and opportunity. Anchored forms encountered a substrate less predictable in firmness and composition. At the same time, increased nutrient exchange may have supported higher productivity in certain zones. Mobility within sediment also allowed burrowers to exploit resources unavailable to strictly surface-dwelling species, expanding the dimensionality of marine ecosystems from 2 to 3 dimensions. The first burrowers thus represent a turning point, not marked by sudden spectacle, but by gradual restructuring. Their quiet excavation altered chemistry, redistributed matter, and diminished the dominance of microbial carpets that had shaped earlier environments. In the wake of these changes, the seafloor became a more complex and responsive landscape, its layers intertwined by living motion that extended below the surface rather than remaining confined above it. At first, they appear as faint rings and narrow cones embedded within the sediment, small structures that resist compression more firmly than the surrounding rock. Unlike the flattened impressions of earlier soft-bodied forms, these remains retain a suggestion of three-dimensional shape. In late Ediacaran strata, mineralized tubes and simple shells begin to enter the fossil record, marking a subtle yet profound shift in how organisms interacted with their environment. Tissue alone no longer defined the limits of the body. Minerals, drawn from seawater, became part of living architecture. These earliest hard parts were modest in scale and construction. Tubes composed of calcium, carbonate, or other mineral phases formed protective encasements around soft internal tissues. Some were gently curved, others straight and hollow, anchored upright in sediment or lying partially buried along the seafloor. Shell-like elements, still simple compared to later molluscan forms, offered rigidity where there had once been only flexible tissue. The ability to precipitate minerals within or around the body required physiological control over chemical gradients, directing ions into crystalline arrangements rather than allowing them to disperse freely. Ocean chemistry played a crucial role in enabling this development. As oxygen levels rose and weathering on continents delivered increased concentrations of calcium and other ions to the sea, conditions favored the precipitation of certain minerals. The saturation state of carbonate in shallow waters likely fluctuated in ways that made biomineralization more energetically feasible. Organisms capable of regulating internal pH and concentrating ions could harness these conditions to construct durable frameworks. What had once precipitated abiotically in certain settings now became incorporated into living forms with controlled precision. The emergence of hard parts also corresponds with rising ecological complexity. As burrowing and movement intensified, interactions among organisms grew more varied. Mineralized tubes may have provided stability within shifting sediments, anchoring bodies against disturbance. They could also have served as deterrents against mechanical damage, whether from environmental stress or from increasingly interactive neighbors. Even in the absence of widespread predation, physical protection would have conferred advantages in habitats experiencing greater substrate disruption. Biomineralization imposed new metabolic demands. Extracting ions from seawater, transporting them to specific tissues, and orchestrating crystal growth required energy and regulatory mechanisms beyond those needed for purely soft bodies. The increasing availability of oxygen supported these costs, supplying the ATP necessary for active transport and structural synthesis. In turn, hard parts enabled organisms to alter their physical presence within ecosystems. Rigid tubes projected upward from the seafloor, modifying flow patterns in their immediate surroundings. Clusters of mineralized forms may have created localized microhabitats, influencing sediment deposition and nutrient distribution. These structures were still rare compared to the abundance of soft-bodied organisms, yet their appearance signals a decisive experiment in durability. Hard parts enhanced preservation potential, leaving clearer traces in the fossil record than earlier forms. They also foreshadowed a broader transformation soon to unfold, in which skeletal frameworks would become commonplace. For now, the seafloor hosted a mixture of old and new architectures, pliant tissues resting alongside emerging mineral reinforcements. Within this evolving marine landscape, the incorporation of stone into biology represented both opportunity and adaptation. Ocean chemistry provided the ingredients. Rising metabolic capacity supplied the means. Ecological shifts created the context. From these converging conditions arose the first persistent skeletal elements, quiet innovations that reshaped the relationship between living tissue and the mineral world that surrounded it. In layers of stone laid down toward the end of the Ediacaran, familiar impressions grow scarce. The quilted ovals, the branching fronds, the soft outlines pressed gently into ancient sediment begin to thin, then fade. By around 541 million years ago, many of the forms that had defined these quiet seas are no longer present in the record. The surfaces that once carried their imprints become dominated by different traces, different architectures of life. The disappearance did not occur as a single visible catastrophe etched into rock. There is no global ash layer neatly separating one world from the next, no uniform signal of abrupt extinction preserved everywhere at once. Instead, the transition appears uneven, stretched across regions and environments. In some basins, Ediacaran organisms persist longer. In others, they vanish earlier. This patchwork pattern suggests a process woven from multiple threads rather than a solitary cause. Environmental change likely played a role. Shifts in ocean chemistry, fluctuations in oxygen concentrations, and alterations in nutrient cycling would have affected communities adapted to stable, low disturbance conditions. Rising levels of oxygen in surface waters may have favored organisms with higher metabolic demands, while deeper zones experienced variable oxygenation. Changes in sediment structure, influenced by increasing burrowing activity, transformed the seafloor itself. Microbial mats that once stabilized sediments and provided a consistent substrate began to fragment under persistent disruption. At the same time, new forms of interaction emerged. organisms capable of more active movement, more directed feeding, and perhaps early predation entered ecosystems that had long been governed by passive strategies. Even without widespread evidence of direct attacks on Ediacaran bodies, competition for space and nutrients could have intensified. Faster growing or more flexible organisms may have outcompeted slower, surface bound forms. In environments where substrates were churned and oxygen penetrated more deeply into sediment, previous adaptations may have become liabilities. The Ediacaran organisms themselves were built for a world defined by microbial dominance and relatively simple ecological networks. Many lacked clear digestive tracts or specialized tissues for complex processing. Their bodies often relied on broad surface areas in contact with sediment and water, absorbing dissolved nutrients or interacting directly with microbial films. As ecological structures grew more layered and dynamic, these strategies may have yielded diminishing returns. Climate variability during the late Neoproterozoic also introduced additional pressures. Oscillations in temperature and sea level would have reshaped shallow marine habitats, fragmenting populations and altering circulation patterns. Such changes rarely leave singular dramatic signatures, yet over extended intervals they can erode resilience. When multiple stressors converge, even gradual shifts may culminate in widespread decline. By the dawn of the Cambrian, the fossil record presents a distinctly different cast of characters. Hard parts become more common, trace fossils grow more elaborate, and body plans exhibit new degrees of organization. The absence of many Ediacaran forms marks not only an ending, but a reconfiguration of life's possibilities. Their disappearance opened ecological space into which new lineages expanded, reshaping food webs and physical structures across the seafloor. The quiet fading of the Ediacaran biota illustrates that extinction is not always explosive. It can unfold through subtle imbalances, through incremental, environmental, and biological shifts that slowly redraw the boundaries of survival. In the stone archive of these seas, the once abundant forms recede, leaving behind an interval of transition in which one long experiment in living gradually gives way to another. Energy began to move differently through the water. What had long circulated at measured, almost unhurried rates now passed with greater intensity from cell to cell, from body to body. As oxygen concentrations rose to more stable and widespread levels, respiration yielded larger returns. The chemistry of metabolism shifted from minimal survival to surplus capacity, allowing organisms to allocate energy toward growth, movement, and interaction rather than mere maintenance. Aerobic respiration extracts far more energy from organic molecules than anaerobic pathways. With greater access to oxygen dissolved in shallow seas, cells could sustain higher rates of ATP production. Mitochondria, already present within eukaryotic cells, operated under fewer constraints, supporting more complex internal organization. Tissues thickened. Body dimensions increased beyond the narrow limits imposed by diffusion alone. Internal transport systems, even in simple forms, became more advantageous as metabolic demand intensified. This increase in available energy altered ecological tempo. Nutrient processing accelerated as organisms consumed, transformed, and redistributed organic matter more rapidly. Where earlier ecosystems had depended heavily on passive absorption and slow filtration, new strategies emerged that required sustained muscular contraction and coordinated behavior. Movement across sediment surfaces became more deliberate, less random. The seafloor, once dominated by stable films and lightly impressed bodies, experienced more frequent disturbance. As activity levels rose, interactions grew more intricate. Organisms that could reach food sources more efficiently or reposition themselves in response to local conditions gained advantages. Energy allowed experimentation with sensory structures, primitive nervous coordination, and directional locomotion. These features did not appear fully formed, yet incremental changes accumulated. The threshold crossed was not merely chemical, but ecological, marking a departure from static communities toward increasingly dynamic ones. Predation likely began to surface within this energetic landscape. Even limited evidence of direct consumption implies a profound restructuring of relationships. When one organism obtains energy by consuming another, rather than relying solely on dissolved nutrients or suspended particles, selection pressures intensify. Defense, evasion, and detection become relevant forces. The emergence of mineralized elements in some lineages may reflect this changing context, providing reinforcement against mechanical damage in a world where contact grew more consequential. Rising oxygen also influenced ocean structure. Enhanced ventilation of surface waters supported higher densities of metabolically active organisms, while fluctuations in deeper layers created gradients that shaped distribution patterns. Regions with sufficient oxygen could sustain faster turnover of organic carbon, accelerating nutrient cycles. Such feedbacks reinforced ecological complexity, promoting diversification of roles within communities. The overall effect was not an abrupt surge, but a gradual quickening. Growth rates shortened relative to geological timescales, generation intervals tightened. Subtle advantages compounded across populations, amplifying small innovations. Energy, once a limiting currency tightly rationed by environmental chemistry, became more readily accessible in certain settings, enabling experimentation with body plans and behaviors that would have been untenable under previous constraints. Within this transforming marine realm, ecosystems began to resemble networks of exchange rather than isolated assemblages. Matter and energy flowed through increasingly layered pathways, connecting producers, consumers, and decomposers in more intricate loops. Oxygen functioned not only as a molecule but as a catalyst for possibility, expanding the range of viable strategies. The seafloor and water column no longer hosted exclusively slow, surface-bound forms. Instead, a gradual acceleration took hold, subtle yet persistent. In this energetic shift lay the foundation for more assertive modes of life, for interactions that would define subsequent chapters of biological history. The chemistry of the atmosphere and oceans had altered the rhythm of existence, and life responded by moving, feeding, and transforming with renewed momentum. Beneath the surface calm of late Precambrian seas, information itself was accumulating, not visible in stone, not etched into sediment, but carried within strands of nucleic acid, coiling inside cells that had already begun to diversify in form and behavior. As ecological interactions intensified and energy flowed more freely, genomes grew in complexity, acquiring new regulatory elements and refining older ones. Evolution was no longer only a matter of survival within fixed possibilities. It was expanding the very toolkit from which bodies could be built. Gene duplication played a quiet but decisive role in this expansion. When segments of DNA were copied, redundant sequences provided raw material for innovation. One copy could maintain essential function, while another drifted, accumulating changes that sometimes produced new capabilities. Over extended spans of time, these duplications allowed networks of genes to grow more intricate, coordinating the development of tissues and spatial patterns within multicellular organisms. Among the most significant components of this emerging genetic architecture were developmental genes, such as those belonging to the Hox family. These genes, conserved across many later animal lineages, regulate the spatial organization of body plans along an axis, determining where particular structures form. Evidence suggests that versions of such regulatory systems were already present before the explosive diversification that would follow. Their existence did not guarantee complex anatomies, but they provided a framework capable of generating them. The importance of these genes lay not merely in their presence, but in how they interacted. Regulatory hierarchies allowed certain genes to activate or silence others, producing cascades of controlled differentiation. Cells in one region of a developing organism could follow distinct developmental pathways from those in another region, guided by gradients of gene expression. This capacity for positional information enabled bodies to acquire front and back, top and bottom, internal layering, and external specialization. As these genetic systems matured, the potential range of morphological outcomes widened. Previously simple forms could experiment with segmentation, symmetry breaking, and varied tissue types. The accumulation of regulatory complexity did not immediately manifest as dramatic outward transformation, yet it set the stage for rapid change once ecological and environmental conditions aligned. Rising biological complexity also fostered feedback between genes and ecosystems. Organisms with more elaborate developmental programs could occupy new niches, altering resource distribution and interaction patterns. Those changes, in turn, imposed selective pressures that shaped gene frequencies within populations. Evolutionary tempo, while still measured against geological timescales, began to feel less static. Variation accumulated within lineages, and selection operated on increasingly multifaceted traits. This period can be understood as a threshold state, poised between long stability and imminent innovation. The raw materials for diversified animal life were assembling at the molecular level even before their full expression appeared in the fossil record. Genetic circuits capable of directing organized growth, spatial differentiation, and coordinated function were forming within cells that still inhabited comparatively simple ecosystems. A planet on the edge of change does not always reveal its transformation in a single dramatic sign. Sometimes the shift occurs quietly within invisible code and cellular machinery, awaiting the convergence of opportunity and pressure. In these seas, beneath currents that carried dissolved oxygen and nutrients, evolution was constructing blueprints for forms not yet realized. The stage was being set within genomes, where regulation, duplication, and integration prepared life for a new chapter of structural and ecological complexity. The boundary draws near not as a sharp line in the sea, but as a gathering momentum within it. Layers of sediment laid down at the threshold of the Cambrian reveal a record that grows suddenly crowded, forms multiply, structures become more elaborate. The quiet experimentation of earlier intervals gives way to a visible surge in anatomical variety. In a span brief by geological standards, the fossil record begins to display an array of body plans that had not previously left such durable traces. One of the most striking developments is the proliferation of hard skeletons. Calcium carbonate and calcium phosphate are organized into shells, plates, spines, and articulated elements that lend rigidity and protection. These structures not only defend against mechanical stress and biological interaction, they transform preservation itself. Organisms that once would have dissolved without a trace now endure in stone, their shapes captured with new clarity. The archive thickens, revealing a biosphere that appears to diversify at remarkable speed. Segmentation becomes more common, allowing bodies to bend with coordination and strength. Jointed appendages begin to extend from trunks, enabling directed movement across and above the seafloor. Instead of drifting or resting passively on sediment, many organisms now propel themselves with purpose. Tracks and burrows grow more intricate, cutting through layers that were once largely undisturbed. The substrate is no longer a stable stage, but a landscape of motion. Among the innovations of this period, the emergence of complex eyes stands as a profound shift. Light had long influenced microbial growth and circadian rhythms, yet now specialized organs concentrate and interpret it with increasing precision. Photoreceptive cells, arranged within protective structures, allow organisms to detect contrast, direction, and movement. Vision introduces a new dimension to ecological interaction. Bodies become not only physical entities, but perceiving ones, capable of responding to distant stimuli rather than solely to immediate contact. With perception comes acceleration. Predation intensifies as animals equipped with jaws, grasping appendages, and advanced sensory systems pursue other organisms with measurable intent. Selection pressures compound. Defensive strategies evolve in parallel, from thicker shells to improved mobility. Energy cycles through ecosystems at faster rates, driven by the demands of pursuit and evasion. The tempo of life, once defined by gradual absorption and slow repositioning, shifts toward rapid engagement. Despite the apparent abruptness of this diversification, it rests upon foundations laid over millions of years. Genetic toolkits capable of patterning complex bodies were already in place. Oxygen levels had risen sufficiently to support sustained activity. Ocean chemistry had enabled biomineralization. The convergence of these factors now manifests in a visible expansion of form. New phyla emerge, their fundamental architectures establishing lineages that will persist, diversify, and sometimes disappear across subsequent eras. The Cambrian does not represent the sudden invention of complexity from simplicity, but the unveiling of potential accumulated through preceding chapters. What had been incremental at molecular and ecological scales becomes dramatic when recorded in mineralized skeletons and dense fossil assemblages. The seafloor transforms into a mosaic of niches occupied by creatures that crawl, swim, burrow, and hunt. As this interval unfolds, Earth crosses a threshold in biological organization. Ecosystems grow layered and interconnected, structured by flows of energy that link producers, consumers, and decomposers with increasing intricacy. Motion, perception, and structural reinforcement become central features of life's design. The approach of the Cambrian marks not an isolated event, but the culmination of converging changes that have been gathering strength beneath the surface for ages. Possibility rarely depends on a single cause. It gathers from converging currents, from chemistry, climate, and inheritance, aligning across vast spans of time. By the close of the Precambrian, several of these currents had strengthened enough to support transformations that once would have failed. The diversification unfolding in the early Cambrian was not an isolated spark, but the result of thresholds quietly crossed. Oxygen stands among the most significant of these thresholds. Sustained concentrations in surface oceans permitted aerobic metabolism to operate at higher and more reliable levels. Cells equipped with mitochondria could generate energy efficiently, supporting muscular contraction, sensory processing, and regulated growth. Oxygen also influenced geochemical cycles, stabilizing certain elements in forms accessible to living systems. Its presence reshaped both physiology and environment, creating conditions under which more demanding body plans could function. Nutrient supply contributed its own momentum. Weathering on continents delivered phosphorus, iron, and trace elements to coastal waters. Shallow seas expanding over continental shelves increased the area where sunlight penetrated to the seafloor, promoting primary productivity. Enhanced cycling of organic matter through increasingly complex food webs allowed energy to be redistributed across trophic levels. Where nutrients accumulated and were efficiently recycled, populations could expand, increasing opportunities for variation and selection. Climate stability, relative to earlier extremes, provided continuity. Although fluctuations persisted, the absence of global-scale glaciations allowed marine habitats to remain connected for extended intervals. Stable temperature regimes and sea levels fostered persistent ecological communities in which innovations could spread rather than being repeatedly erased. Continental configurations supported broad epicontinental seas, environments that combined access to nutrients with moderate physical disturbance. Such settings acted as crucibles for diversification. Genetic innovation formed the internal counterpart to these external conditions. Regulatory networks had grown more elaborate through gene duplication and modification. Developmental genes governing axial patterning and tissue differentiation were already embedded within genomes, enabling coordinated construction of complex anatomies. Changes in gene expression, timing, and location could produce significant morphological differences without requiring entirely new genes. This flexibility allowed evolution to explore diverse forms rapidly once ecological opportunity widened. Feedbacks among these factors amplified their effects. Oxygen enhanced metabolic output, which supported more active interactions, which in turn selected for improved sensory and locomotor abilities. Nutrient abundance sustained larger populations, increasing the raw material for mutation and recombination. Stable climates allowed successful body plans to persist and diversify across regions. Genetic systems capable of modular variation translated environmental pressures into anatomical innovation. None of these elements alone would have sufficed. Without oxygen, active predation would have remained energetically constrained. Without nutrient influx, ecological expansion would have stalled. Without relative climatic continuity, new lineages might have disappeared before establishing themselves. Without adaptable developmental toolkits, morphological experimentation would have been limited. Together, however, these conditions formed a scaffold upon which complexity could rise. The early Cambrian diversification thus reflects the intersection of geochemistry, ecology, and genetics. It is an expression of Earth system integration, where atmospheric composition, continental processes, ocean circulation, and molecular evolution converged. Life did not abruptly invent complexity. It grew into it as planetary circumstances permitted and encouraged new strategies. In this light, the rapid emergence of varied animal forms appears less as a sudden mystery and more as an outcome of cumulative preparation. For millions of years, thresholds were approached, crossed, and reinforced. When the necessary ingredients aligned, evolution accelerated, translating long-hidden potential into visible diversity. The planet had assembled the conditions required, and life responded with expansion. For millions of years, the rhythm of life remained measured and subdued. Seafloors were layered with soft impressions and anchored forms that grew slowly, drawing sustenance from surrounding waters without urgency. Interactions unfolded at a pace set by chemistry and diffusion rather than pursuit and escape. The Ediacaran world, though biologically significant, carried an atmosphere of extended equilibrium. Communities were structured, yet restrained, their boundaries defined by gradual environmental gradients and modest ecological exchange. Within that extended calm, Earth's systems were quietly calibrating. Atmospheric composition had shifted toward sustained oxygenation. Ocean chemistry was trending toward conditions that could support mineralized tissues and higher metabolic rates. Continental arrangements promoted shallow marine expanses where light, nutrients, and oxygen overlapped. None of these developments announced themselves as harbingers of upheaval. They accumulated in the background, altering thresholds that governed what life could attempt. The contrast between this long stability and the dynamism soon to follow is striking. In the Ediacaran, bodies largely rested upon sediment surfaces or embedded gently within them. Growth was deliberate, and evidence of aggressive interaction is sparse. Energy moved through ecosystems at steady rates, redistributed primarily through passive absorption and simple feeding strategies. Geological processes continued to reshape coastlines and basins, yet biological tempo remained comparatively restrained. Such prolonged steadiness did not signify stagnation. Beneath the apparent calm, variation persisted. Genetic networks diversified. Subtle anatomical innovations appeared and were tested within local contexts. Environmental oscillations, though less extreme than earlier in Earth's history, introduced selective pressures that refined physiological resilience. The planet was not static, it was preparing, its components gradually aligning. As oxygen concentrations stabilized above critical thresholds, the energetic ceiling that once limited activity lifted. Nutrient delivery from weathering continents sustained productivity along shallow margins. Ocean circulation patterns facilitated oxygen penetration into habitats previously constrained by low availability. Climate patterns, while variable, avoided global-scale disruptions that would have reset ecosystems repeatedly. In this layered convergence, biological potential expanded quietly. The impending shift toward Cambrian dynamism did not arise from sudden invention, but from readiness. Ecological frameworks capable of supporting predation, rapid movement, and complex food webs required stable scaffolding. Long intervals of relative equilibrium provided that support. Populations persisted long enough for incremental genetic changes to accumulate. Developmental systems matured, integrating regulatory pathways that could generate organized body plans when ecological opportunity intensified. When diversification accelerated, it did so against the backdrop of this deep preparation. The fossil record's apparent rapidity reflects not a spontaneous eruption, but the crossing of multiple thresholds at once. The quiet world of the Ediacaran had laid the groundwork by sustaining communities through sustained chemical and climatic calibration. Energy flows, genetic toolkits, and environmental structures were primed. In examining this contrast, it becomes clear that motion often follows silence. Systems approach points of transformation gradually through cumulative adjustments that may be imperceptible within any single generation. The Ediacaran calm represents such an approach, a prolonged interval in which Earth assembled the prerequisites for accelerated biological interaction. When change emerged, it did so upon foundations built during that extended stillness, converting preparation into visible diversification. Thus, the quiet before motion was not empty or inert. It was a phase of alignment where atmosphere, ocean, continent, and genome settled into configurations capable of supporting a new ecological tempo. From this poised state, The planet would soon enter an era defined by interaction, mobility, and complexity, yet its roots lay firmly in the long stability that preceded it. Measured against the full arc of Earth's history, the age of animals occupies only a slender portion. The vast majority of time since the planet formed unfolded without shells clattering across reefs or fins cutting through open water. For billions of years, life was present yet almost entirely microscopic, woven through oceans in films, plumes, and drifting cells. The story that eventually led to forests, reefs, and moving bodies rests upon an earlier foundation that was small in scale yet immense in duration. Microbial communities shaped continents and atmospheres long before complex organisms appeared. Through photosynthesis and respiration, they altered the chemistry of air and sea, transforming iron, cycling sulfur, and stabilizing carbon. Their metabolisms mediated exchanges between rock, water, and sky. In sediments and shallow lagoons, layered mats to recorded seasonal shifts and nutrient flows. These quiet systems persisted across intervals of volcanic upheaval, asteroid impacts, and climatic extremes. Stability did not mean stillness, but continuity through change. Complex ecosystems emerged gradually from this prolonged microbial dominance. Cells joined in colonies, differentiated, and formed bodies with specialized tissues. Genetic networks expanded and refined developmental programs. Oxygen rose to levels that supported energetic demands beyond simple maintenance. Nutrients circulated with greater intensity, and continental arrangements provided diverse habitats. Each step was incremental when viewed against the deep timeline, yet together they redirected life's trajectory. The later proliferation of animals, with movement, perception, and interaction, can be understood as an expression of capacities assembled over immense spans of time. Predation, locomotion, and intricate food webs depended upon oxygenation, nutrient supply, climatic moderation, and genetic regulation that had matured earlier. What appears dramatic within the fossil record rests upon quiet preparation embedded in microscopic persistence. Earth was alive long before it was loud. The oceans carried metabolic flux in silence, unaccompanied by calls or rapid pursuit. Energy flowed through pathways governed by diffusion and chemical gradients. Even as the Cambrian introduced visible dynamism, the majority of planetary history remained characterized by subdued yet relentless biological activity. Microbes continued to dominate numerically and chemically, sustaining cycles upon which larger organisms relied. The silence of those early ages shaped everything that followed. It allowed atmospheric transformation, provided evolutionary experimentation at cellular scales, and maintained resilience during environmental stress. Without prolonged microbial endurance, oxygen would not have accumulated, nor would nutrient cycles have stabilized sufficiently to support complex bodies. The endurance of small, simple life ensured that when thresholds were crossed, the planet was ready. Human existence, like that of every animal, traces its lineage through this extended quiet. Our metabolic pathways echo ancient microbial chemistry. Our cells retain organelles that originated in symbiotic unions between ancestral microbes. The conditions that permit us to breathe and reflect are outcomes of processes set in motion long before vertebrate eyes first opened to the sea. In contemplating the longest chapter, perspective broadens. The animal-dominated world feels expansive from a human viewpoint, yet it occupies only a recent layer in a much deeper archive. Beneath it lies an ocean of time, during which life persisted without spectacle, transforming the planet through patience. That quiet endurance was not empty or insignificant. It was formative. From this vantage, the history of life appears less as a series of sudden bursts and more as a continuum shaped by sustained groundwork. The calm epochs, extending across